to talk to this community. So today what I thought I would do is use the time that I have to give you a little bit of a vignette of what how we use proximity-dependent biotinylation to really set up the stage to begin to understand dynamic cell organization. And this is a fairly recent picture of my lab, and the only point I want to make today is that this work has involved many individuals in the lab over the years to really set up the method. And so just before I go into the presentation itself, I think it's really important to appreciate the role of subcellular localization and in fact, organelle organization in disease. In fact, organelle function or dysfunction underlies multiple diseases. For example, Golgi enzyme aberration is really at the core of congenital disorder of glycosylation, and lysosomal storage disease, such as Gaucher and Tay-Sachs, are also really important. But itself, the structural integrity of the organelle is really important. For example, mutation that underlie progeria disrupt the structure of the lamina, and there's many also myopathies associated with cytoskeleton deregulation. Positioning and trafficking is also really important, and disorders such as Emery-Dreifuss muscular dystrophy are caused by problems with nuclear positioning. And lastly, you can have, of course, individual protein that mislocalize, and the poster child for that is that cystic fibrosis mutation at delta 508, which, if corrected by now a number of different agents, can essentially rescue the function and recover the disease. So really understanding how organelles and how proteins that are associated to organelles is really critical to understanding disease and provide ways to overcome those diseases. However, not all organelles and not all structures are created equal, and there's different type of structural units in eukaryotic cells. Some that are well known to be bounded by membranes, such as the mitochondria, but also some that are formed by phase separation, including the nucleolus and some of the smaller nuclear bodies, and also different cytoskeletal elements such as intermediate filaments shown here. And what's really important to know is that not all of those structures are going to be as easily amenable for biochemical fractionation that has been classically used to uncover the composition of proteins within each of those structures. And so many years ago now, my lab and many others in the world have started using this approach introduced by Kyle Rue and Brian Burke, that are now called collectively proximity-dependent biotinylation, or in this case, BioID, in which a protein of interest is fused to an enzyme. In the case of BioID, it's a biotin ligase that then gets expressed at a near endogenous level in a relevant cell system or organ system. And the basic idea there is that the bait will localize where the endogenous protein should localize, and you can supply a substrate for this enzyme, which is this biotin ligase, that gets activated to a reactive intermediate that then goes and covalently label protein in the vicinity of the bait. And depending on the enzyme that you're going to be using, you're going to need to do this labeling for different amount of times. So for example, with the initial enzyme called BurA, which most of the work that I'm going to show today has been performed, you would need something like 10 plus hours to get a very strong labeling. And this is not the case now with some of the most recent enzymes that have been engineered. And I'm going to highlight two of them today called Mini Turbo and Ultra ID. But the basic idea is that once you have this labeling, you don't need to maintain interaction or organelles intact. And in fact, you can lyse cells under very harsh conditions that are going to be breaking down all of those organelles protein complexes, and in fact, partially denature the proteins. But the basic idea is you have now created this permanent mark, which is biotin, onto the protein that were in the vicinity of the bait in the living cells. And you can capture those biotinated proteins through streptavidin affinity chromatography. And this can be followed by elution using enzyme. We used trypsin in our lab, and identification and quantification by mass spectrometry approaches. The only point that I want to make here is that you've now expressed inside cells a active biotin ligase. It's going to generate clouds of biotin, and you need to design very careful controls to be able to untangle which of the protein that you detect in your bioID experiments are seen because they were really in a specific way in proximity of the bait versus proteins that are abundant and easily biotinylated by any biotin ligase. And we do that by designing parallel set of control that we include in all the experiments and using your statistical tool, particularly this SAINT tool, to untangle what is a true interactor and what is a contaminant. So the beauty of this system is we've now separated the detection of a proximity from the identification of what was in proximity. And we and others have now demonstrated that it works in most organelles, both membraneless and membrane-bound, and that we could use a lot of the same tools that we had previously developed for affinity purification coupled to mass spectrometry to untangle the signatures. But the thing that we see with every tagging approach is that the tagging in the bait expression may not always be neutral. And sometimes stepping back a little bit and use bioID to report on endogenous protein might be a good idea. And I just want to tell you a little bit how we've been doing that in our lab. So what we've been applying as a principle is that the association inside living cells between those untagged protein that we're going to be called prey is something that is informative. So imagine that you have those four balls that are each a prey. So this is an endogenous protein expressed in a cell at a certain level and localized to a certain localization. If we do a bioID experiment with a specific bait shown here by that concentric ring, what you're going to be able to see is a relationship between this bait and all of the preys inside the cells. And this can give you some quantitative assessment. If you do that only one time, you cannot really tell anything about the relative proximity of the preys to one another. But if you do that multiple times, shown here by different baits, each generating their concentric rings, what you can see is your quantitative matrix here at the bottom is now providing information that can directly inform on the relationship, a spatial relationship between some of the preys, in which case here, the yellow protein and the dark gray protein are in very, very close proximity and are picked up always the same relative level by each of those baits. So you can really use that at a larger scale to really reconstruct the relationship between those endogenous and not tagged protein. And this really can help creating what we call cell map and organelle map. And I will give you a few examples in the next few slides. So I just want to highlight, we published on that many times. The first paper in which we did that was a collaboration with Brian Roth and Laurence Pelletier, looking at the centrosome cilium interface, looking at the proteins that were recruited to these very important organelles. We also did that for a number of membrane bound structure, including mitochondria with Eric Shoebridge. But I just want to highlight this particular study led by a former postdoc in my lab, Dr. Ji-Young Yoon, that really set the stage for how we think about data analysis for these particular type of questions. And so what Ji-Young was interested in was to define the proteome of Peabodase, which are highlighted here in these green structure, and stress granule, which is an adjacent structure shown here in red on those microscopy image. And as you can see, those are very relatively small, heterogeneous structures, are membraneless, and they're associated with RNA biology. And they both are in fairly close proximity to one another. So really defining the proteome in a specific manner for each of those organelles was problematic. So what Ji-Young and Wade Dunham at the time did is they put bioID markers on several of the Peabodase and several of the stress granule components and started doing proximity-dependent biotinylation analysis, and really realized that this was nice and could, have a hint of untangling the structure. To get that to a much larger scale, however, we felt that we needed to map throughout the path of synthesis and degradation of the mRNA so that we could really tell what is the specific interaction or proximal interactome of each of those structures. And what you see here is essentially the result of about 122 base. Actually, it's 140 base, but coding for 122 unique genes. That really helped us define using computational approaches such as non-negative matrix vectorization, as well as Pearson correlation, the composition of stress granule and their adjacent component, the P-body. We also were able to hint through this particular work to the structures inside the nucleus, including the nucleolus itself, and a number of different structures that we now recognize to be nuclear bodies. So really the point here is that if you do those experiments in high density, you can get very, very fine resolution to the composition of organelles, including membraneless organelles. A current student in the lab, Boris Yakov, has now taken this a little bit further to look again at the composition of all of the nuclear bodies that we could analyze, and that now includes structures including the nuclear speckle as well as the paraspeckle and other nuclear body, and this is now under review. So this is telling you that we can use this approach for single organelles or a small collection of organelles, but what about looking at the entire cell? And so this was done in the first generation by a former student, Chris Go, and computational expert somehow using a pipette, James Knight. And the basic idea was if we were able to cover the cell with well-selected markers of organelles, this came from annotation of cell biology classical organellar marker. And if we profile many of them inside the cells and put some quality matrix, we could maybe be able to generate an entire map of the human cells. And so we did that by generating 244 baits across 25 subcompartments that really enable us at the end of the day to report on the proximal interaction from 192 of those baits and about 40% of the expressed proteome that was done in 293 cells at steady state. And to get to really untangling the structure of the data, we were reporting on 36,000 high confidence proximal interaction using our Saint analysis. What that really told us is that if we do this kind of prey-prey relationship analysis, that really told us that we were easily able to uncover the different large compartments, such as the nucleus, the cytoskeleton, mitochondria, and a number of different membranes, as well as soluble complex inside the cytoskeleton. But even within those structures, we had sub-organelle resolution. For example, in the nucleus, we could easily see components that belong to the nucleolus and other of those nuclear bodies. So this is really powerful, and it really helped us really defining new localization for very poorly characterized protein, and we had a really high validation rate, at least 80% validation rate for the new localization prediction, and we were also able to find new components of membrane contact site that we could validate functionally. But the problem with this particular approach is the way that we went at it was actually a little bit cumbersome, and I'll get back to that in a minute. So at the end of the day, this particular effort gave us a bioID toolbox. So we have a really nice website that enables people to explore this proximity map of the proteome, and also use that as a resource to analyze your own data. And we develop, in part through the cell map effort, but also some of the other project, a resource for visualization of interaction and proximal interaction data, as well as new tools that help us, now that we generate a lot of data really quickly, to really provide annotation to each of the protein that we have through using API to a number of different and very highly valued databases. But again, the problem with the current map is it is static, right? It is only done into 93 cells, and this was all done after us stably integrating each of the bait inside the cell. So it's really time consuming. Going across multiple cell lines for that would not necessarily be feasible. Again, this map is static. We just did it under standard growing condition. And of course, it's really out of date because it's based on older instrument and this initial enzyme that required long labeling time. And quantitatively, this is not the greatest map because we were using spectral count as a proxy of abundance at the time. So what we want to do is to really enable us to move way beyond this and generate not one cell map, but multiple cell map. Some of them would be telling us a little bit about the cell type specificity. Some of them will tell you about short dynamics. For example, they can be stress, stimulation, or cell cycle. Some of them might be really important to look at long time of dynamics, for example, reprogramming and differentiation. And some can be a support for personalized medicine, for example, looking at patient-derived cells. But if we were to do something to the scale of the current initial cell map, we would have to have 200 baits, 600 sample per map. So we have a really huge need to scale up this process. And so how can we do that? So first, we can computationally try to estimate how many baits you really need to be able to do this kind of exercise, right? Do you need the full 200 baits, or can you get away with much fewer? And so Vezal Kasmei-Far, together with Saya Sedihi in the lab, who I forgot to list on this particular map, but she's on the bioarchive, has come up with a way to essentially estimate how many baits we need to cover as many of the praise, but to also keep the same structure of the network. And so Vesal came up with a new tool that he called GeneBait that's based on a genetic algorithm that helps select the number of minimum number of baits that can recapitulate all of the different structures that we were able to see in our initial map. And this is actually good because it was able to define that, at least in this data set, we were able to maintain the accuracy and the structure with only 60 baits rather than the 200, which is more than a threefold reduction. And this is true for other data sets that we've looked at, including this RNA body data set for Geong. We could really reduce it to much fewer baits. So we think that this is good. We don't need to look at all of the baits all the time. So this is already a good step. But we also need to get beyond this need to generate stable cell lines for everything. And so the strategy that we selected is based on an approach that former postdoc Peyman Samavarshi-Tirani has developed and really finely tuned based on a lentivirus delivery system that is regulatable by doxycycline and also is compatible with a number of different marker selection strategy. The big advantage of the system is that we can generate a big pool of virus and really go horizontally across different cell types. And we've demonstrated that this is nicely tuned and can be regulatable. And the basic idea too is we can now freeze those viruses. And the next question became, which enzyme should we use, right? If we move to CellMap V2, we know we want to go into dynamics, both kind of longer time frame and also short labeling time. So which of the enzyme can we use, right? So which enzyme that is now, there's at least 10 that have been published, which is fast enough, which has a low enough background, which is able to respond really well to exogenous biotin and give us these early time point temporal control. But more importantly, can we have enzymes that work well across as many compartments as possible? And so Saya and Kozar, together with many people in the lab, have started really looking at that systematically. So there's now, as I said, more than 10 published bioenzymes that are derived from 3 bacterial species, E. coli, this thermophile called Echifex aeoleucus, and also Bacillus subtilis. And so which one will we use will be important. So what we decided to do is to put each of them, both as N-terminally and C-terminally tag fusion, in the same backbone that we use for the cell map. So we're generating stable HEK293 flip-in. And we're going to be putting in all of them laminate, which is actually the first enzyme that was used to define this bio-ID. So we know it's going to work really well. And as a negative control, we use empty cells, but we also use this GFP protein, green fluorescent protein, which is expressed in at least as high of a level as laminate. And the basic idea is using this flip-in system is we can control the level of expression of the base so that we make sure that we compare apples to apples. And to make a long story short, where we want to be is in that star corner where we get a very strong biotin signal in as little time as possible so that we can do those dynamic study. But we also need to have a good response to exogenous biotin. So in the absence of biotin, we don't want signal, and we want signal only when we add this exogenous biotin. And so I can already tell you that some of the enzymes that we being Saya, Kozar, Rod, Rawan, some of the enzymes that they tested were really not sensitive enough. This include the first generation enzymes as well as some of the second generation, but not optimized. We had at least one enzyme that we couldn't find any significant response to exogenous biotin, so we deleted it as well. Turbo-ID is an enzyme that was developed through by enlisting through a number of directed evolution. It's extremely the strongest that we've seen. However, it's very hard to get a clean baseline for it. So if you need to have it for very fast dynamic study, this is actually kind of problematic. And then we had a number of enzymes that fell in between, with two of them in particular, Ultra ID and Mini Turbo, hitting what we think is a Goldilocks spot, meaning that they have a very nice signal intensity. They can label in a very short amount of time, but they also respond really well to exogenous biotin. And so the next question became, can they be specific? And I'm just going to show you one figure. This one is actually from an ELA cell study that Kozar Vafai has been doing in the lab. And so what you see in this is all pairs of 8 different baits that Kozar has tagged with either Mini Turbo or Ultra ID. And what you can appreciate, first of all, is that by and large, the bait cluster with one another, right? So it's not like the proximal interactome that you get in both of them is very, very different. But in many cases, we have a different and in many cases a better labeling with Ultra ID compared to Mini Turbo. And when we look at the specificity, what we notice is even in the ones such as RAB5, where the labeling was fairly similar, Ultra ID seemed to recover more specific prey, while Mini Turbo recovered chaperonin. And this was even more pronounced in the case of some of the cytoskeletal baits, such as VASP1 here, but also PFN1, where the Ultra ID specific protein were actually mislocalized to the mitochondria. So we feel that after all of these analysis and similar analysis that Saya did in 293 cell, that while Turbo ID is the fastest and that we still use it whenever we need maximum sensitivity, that Mini Turbo can get us a clean baseline, and it works with many, if not most cases, but we found some exception where it didn't work. By contrast, we find that Ultra ID, which was developed in Europe by Julian Bethune, is at least as good and often better and sometimes much, much better than Mini Turbo in all the cases that we've seen so far. So we haven't seen any case so far where Mini Turbo clearly outperformed Ultra ID. So a couple of notes on this. It's smaller. It's 19 kilodalton. It's also from a thermophile, so we think the folding is actually a little bit easier because of that. And it seems to have less trafficking issue than Mini Turbo. So we now have decided that for the cell map V2, this is essentially the enzyme that we're going to be using. The only point that I want to make is the background labeling by each enzyme is quite distinct, and related enzymes have the same background. So we will have to take that into consideration when scoring against Saint. So we cannot use a Mini Turbo data set to score Ultra ID data. We need to have proper matching control, and we will be redesigning some of our own tools, including a contaminant repository for affinity purification, and coming up with maybe an enzyme selection tool to really account for this background. So now we have reduced the number of bait. We also have selected a different enzyme. I told you that we have this lentivirus delivery system into which several of the people in my lab have been now cloning those faster acting enzyme. We also, and I'm not going to talk about that too much today, we've also improved our mass spec acquisition pipeline that enable us to go from, at the time, about 4 samples a day in protein interaction data to now something closer to 60 samples a day. So this throughput is really important for us. We've been playing with quantitative accuracy through integrating data independent acquisition, as well as a sample prep for improving sensitivity. So we now have a pipeline that is actually a little bit more feasible for doing this new version of the cell map, which I'm not going to tell you too much about today because we're still working on that. But I want to show you just before I close a couple of the vignettes of where we're going and the kind of studies that are enabling this cell map. So first, we just released, and I'm not going to tell you too much about that because this is published and I should have updated that. It's now in Science Signaling. Dr. Rasha Al-Mismar has now published a feasible extracellular turbo ID method that enables us to look outside the cell and not only intracellularly. And this really enables us to look at the reorganization of the extracellular side of the plasma membrane following, for example, the ligand or any kind of treatment. So this is something that could be useful for many of you. But the pilot project to look at dynamic came from 2 different sets of studies, and this is why we think that this cell map will work. The first type of project was really looking at a reprogramming, so take somatic cell reprogramming, which really is a slow kinetic project. And the fast kinetic project that we look at is EGF stimulation. Basically, this is a bit different, and it required us to think about different parameters that we will need to include in our cell map V2. So for example, for reprogramming is so kinetic, so, we need to make sure that we will be adjusting for protein expression, because as cells reprogram, the entire proteome, in fact, the transcriptome, but importantly, the proteome is changing, and really figuring out whether we have a change in the proximity biotinylation because the proteome has changed or because something specific happened to change the organization of the proteome is really important. Importantly, this really told us that we needed to optimize our lentiviral pipeline. And the successes and failure of that really told us that the cytoskeleton and membrane reorganization were actually a difficult spot for bioAD experiment, which is why we wanted to test this Ultra ID versus mini turbo on cytoskeletal element, because we felt that this was actually a bit challenging. So in the case of EGF simulation and fast kinetic, and this is again just a cartoon that shows that once EGF, the epidermal growth factor, sees the ligand, it induces ligand dimerization, but also a rapid internalization of the ligand that can then be either recycled or degraded. And the time course for that are very fast. Some of the events of the initial internalization happen within the order of 10 minutes, and really being able to do a time resolve analysis was really important. So this really forced Claire to develop ways to look at minimum labeling time and really a multi-bait approach to really understand the signaling proximal to this EGFR receptor. It also forced us to really think about dynamic visualization. And we had introduced in a science paper led by Jeff Esket many years ago now, the idea that we could couple bioID with CRISPR-mediated depletion to really map the ordered events in a signaling pathway. And I won't go into too much detail. This is just some of the data that Claire has been getting. So she's able to really do 2 minutes labeling, kinetics for early phases of the stimulation. And then after that, she overlaid that with a 15 minutes kinetic. And she's really able to see the epidermal growth factor as a bait as it progresses through this particular pathway, really revealing a cluster of organization that really map really nicely to this endosomal recycling and restoration pathway. So we think now that thanks to the work of these people, Claire and the other people, that we are really well positioned to really tackle these cell mapping efforts. And just a couple of notes of where we're going. And this is just to come back to the presentation that Hugo gave you a few months ago, I think now. So we're really looking at signaling condensate. We're really looking at signal integration in the context of aberrant signaling. For example, in the context of translocation, Hugo is really trying to map whether these dynamic condensate that occur following this translocation are a common event, and whether there is specificity that is acquired. And this is all employing some of the same technologies that I showed you. And I just want to say that we're not only doing bio-ID in BioLab, although we mostly do bio-ID, but we're also recently coupled our effort in mass spectrometry with the effort of colleagues here in Toronto that are doing in vivo CRISPR screen on the backbone of common oncogenes, such as RAS G12D in lung cancer, to look whether the combination of different aberrations, so it can be either the overexpression or downregulation of tumor suppressor and oncogene, affect the proteome of tumors derived from this G12D model, either by changing the proteome within the tumor or the tumor microenvironment. And so this is actually extremely powerful. We already have amazing data that really indicates that we can uncouple the genotype to phenotype processes in the context of lung cancer, and we'll be looking for new people for this. So I'm going to stop here. I'm out of time. I just want to acknowledge all the people in the lab that have done the work. I've mentioned several of them, true, but this also includes several of the former lab members and collaborators, as well as funding. And I'll be happy to take some of your questions. Thank you very much.